Hello everyone, my name is Suarez Castellanos and I'd like to thank you for the opportunity to come and present to you our work titled Spatial Temporal Description of Electrophysiological Responses Stimulated by Focused Ultrasound using Microelectroarrays, or MEAs for short. This is a collaborative work between the Laboratory in Lyon, France and the Collège de France in Paris, France. And the general objective of this study was to design and build a hybrid platform that would integrate a focused ultrasound neurostimulation system with a microelectroarray recording system for the study of the electrophysiological activity produced by focused ultrasound on hippocampal brain slices. And we use this neural model because it is a model that has been widely studied and described in literature for studies of synaptic transmission along major neural pathways of the hippocampus, such as Schaefer collaterals and the dendrite gyrus. And at the top of the poster, we have a description of the system that was designed and built for this purpose. And on the left, we have a picture of the actual setup along with some of the focused ultrasound parameters that were used to stimulate these neural structures. We used a central frequency of 1.78 megahertz and very short pulses of 160 microseconds applied at a pulse repetition frequency between 1 and 5 hertz and applied with at relatively high pressures at the focus ranging from 3 to 7 megapascals. We use these high pressures because we wanted to measure the causal response produced by short single pulses of ultrasound as opposed to what's been described in literature by other teams that tend to use lower pressures applied in the form of repetitive pulses or pulse trains. To the right of this picture, we have a schematic of the setup that was built for this purpose, along with pictures showing the microelectroarray chip and the hippocampal brain slices and how this neural structure is placed with respect to the to the array of microelectrodes contained within the MEA chip. In the results section immediately below, to the left, we have a direct comparison of the responses generated by focused ultrasound pulses as compared to the responses generated by pulses of electrical stimulation. The responses generated by focused ultrasound are shown in red as averages of the responses generated by consecutive ultrasound pulses, and the pink shows the standard deviation of these consecutive responses. And similarly, in blue, we have the average response produced by consecutive electrical stimulation pulses, and the lighter blue shows its standard deviation. And we can see by comparing the two that both responses exhibit similarities in that they both start with a fiber volley, which corresponds to the arrival of the presynaptic signal or the arrival of the electrical signal to the synaptic terminal. This response is immediately followed by what is known as an excitatory postsynaptic potential, which corresponds to the transmission of this presynaptic signal to the postsynaptic neuron. To the right of the results section, we have a study consisting of a spatial reconstruction of the signals recorded by all 60 electrodes within the MEA chip. Using this reconstruction, we were able to measure and study the spatial distribution of the responses generated by single focused ultrasound pulses. And we were able to use this analysis to not only study the spatial characteristics of the response, but also the temporal characteristics of the response with high temporal resolution, since the sampling rate of the system was 50 kilohertz. And using in this schematic, we can see that the highest responses were recorded in towards major neural pathways of the hippocampus, in the dendrite gyrus in the top right corner of the array, as well as the CA3 region of the Schaefer collaterals. In conclusion, this study demonstrates that hybrid systems that integrate focused ultrasound stimulation systems and microelectroarrays are viable tools that could be potentially used for further studies of the biomechanisms involved in the electrophysiological activity stimulated by focused ultrasound neurostimulation. Thank you for your time, and I will now take questions from the audience.